All right. So while I have most of you as a captive audience, since you just took your exam and most of you are here, remember that your draft term paper is due towards the end of the month. Your draft term paper will be assessed by your peers, and so they will be scoring you based on the same rubric that I will use. So if your draft term paper is literally a sentence that says, "My disease is X," you will probably do very poorly on your peer assessment. So your draft term paper needs to be as complete a term paper as possible if you would like the most points possible. So remember, this has been assigned since the beginning of the semester. You theoretically should be working on it. You're probably not, but maybe you will now. And you can probably still get a really solid draft if you start now and begin working. Okay, so that's just my friendly advice to you is don't wait until October 24th or whatever to start working on your draft. Because when your peers give you a poor grade, which they will, I won't be able to help you. Okay, so you want it to be as complete as possible. Because then you have the opportunity to go back and fix anything they pointed out, anything you noticed before I read your final draft. So let's continue then talking about biofilms. We left off Monday talking about Pseudomonas aeruginosa, which is one of my personal favorites. It's a pretty gnarly bacterium. It causes a lot of significant complications. It's definitely acquired in hospital settings, so it's something to be concerned about for those of you who ever plan to go to the hospital. Okay. So Pseudomonas aeruginosa, they do quorum sensing. So they communicate using quorum sensing molecules, and for them to form a biofilm, they do have to be able to do quorum sensing. And so they accumulate their particular quorum sensing molecule, which in this case is one of the acyl homoserine lactones, and that helps to signal to them that the population is growing. Okay. Remember, that's what quorum sensing is for. We have other members of the same species, and there are either very few of them or a lot of them. That's how they can tell population density. The accumulation of these quorum sensing molecules is what regulates the gene expression that will lead to biofilm formation. So in Pseudomonas, when quorum sensing molecules accumulate, they will turn on the genes necessary for the secretion of the polysaccharides to build the biofilm, and also of that signaling molecule, cyclic di-GMP. Remember, cyclic di-GMP is important for biofilm formation. It helps turn off motility, turn on adhesion, turn on polysaccharide secretion. Okay? So you get the initiation of extra polysaccharide production, and you get the downregulation of the flagellum. We're not moving anymore. We're going to sit here and build our biofilm. Okay. So in Pseudomonas aeruginosa, they begin to form a biofilm when there are enough of them. So when they begin to sense enough of this quorum sensing molecule. Otherwise, they're usually going to move around. They're highly motile, and they want to find the best environment. So when they find the best environment, then they'll sit down and perhaps begin forming a biofilm. In Vibrio, we see something a little bit different. In Vibrio cholera, we also form biofilms. However, unlike in Pseudomonas aeruginosa, in Vibrio cholera, we form biofilms at low densities. So they use inter and intracellular signaling to control biofilm formation. They use cyclic GMP to regulate those genes, so the turning off of the flagellum, secretion of the polysaccharide. But in this case, Vibrio cholera forms biofilms when they are at low densities. And they usually will do this when they're found in their natural environment. When there aren't enough nutrients, they'll probably sit down and form a biofilm and hope that they can protect themselves. So they can attach to marine surfaces for better access to nutrients and protection. And those surfaces can include things like plankton and crustaceans, so other animals. or it can include marine sediments. So this is why it can be really dangerous to consume undercooked shellfish from waters that are known to carry Vibrio species, because these shellfish are home to Vibrio. So undercooked shellfish from contaminated waters can give you cholera or some of the other Vibrio infections. Okay, cold water, you're probably safe. Warm water, don't trust it. And so again, what Vibrio is trying to do is, remember, the way that Vibrio cholera causes most severe disease is by secretion of toxin that leads to diarrhea. But they don't want to be flushed out of the system until there are enough of them. So when they are at low densities, they'll form biofilms, right? They'll get to really high densities, secrete their toxins, and detach. So once they get to high densities, they no longer need to be in a biofilm. They're ready to go. So at high densities, they won't form their biofilm anymore, and they'll leave. So any questions on biofilm formation before we begin talking about how microbial cells can regulate their interactions with antibiotics?
So would boiling any shellfish kill the cholera?
Oh yeah, cholera are pretty sensitive to high heat. So if you properly cook your food, no problem. Yeah, boiling your water. I don't know what it's like. Lord of the Flies in here today. It's that time of year where this classroom gets full of flies. I mean, they cleaned out trash corner. Somebody was like keeping all their trash in that corner, so I don't know. So sorry about the flies. It's more embarrassing when we bring outside speakers and the outside speaker is like, they're like, you guys can deal with the flies. I guess the outside speakers don't have to deal with the flies. Any other questions about biofilms? So again, biofilms are a really important mechanism, of course, for causing human infection, but they also are really important in microbes that don't cause human infection. So they can be very important in environmental settings and for protection from predation. Lots of things eat bacteria. For example, amoeba tend to eat bacteria. They call it grazing. The amoeba like graze on bacteria. But if they form a biofilm, if the bacteria form a biofilm, it's much more difficult for the amoeba to phagocytize cells. So it can provide protection from their own predators as well. Okay, so in this section, we need to talk about antibiotics. And so this is the section that's going to be tightly related to what you're doing in the lab. So antibiotics were first discovered as compounds produced by one microbe that can kill another. And so the first antibiotic that was really discovered, purified, marketed is penicillin. And penicillin is produced by a fungus, Penicillium. And it was discovered because it can inhibit the growth of Staphylococcus. So I'm going to tell you about a really poor laboratory practice. This is one of those things that you probably shouldn't do it, but at the end of the day, the guy who did it won a Nobel Prize. So apparently in Europe, it's really common that everybody just goes on vacation in August, like the whole month of August. Super jealous. I'd love a whole month of vacation. So Alexander Fleming was growing Staphylococcus in his lab. He and his lab tech went on vacation in the month of August, and they left their Staphylococcus plates on the lab bench. When they came back in September, the plates were all contaminated with mold because that's what happens. And they saw that in places where the mold had grown, the bacteria had not. So it was an accident. It was improper lab hygiene. And he won a Nobel Prize for the discovery of penicillin. I would argue that along with vaccines, really the development of antibiotics is what has improved human lifespan. You could argue that it helped us win World War II because we had antibiotics and the Germans didn't. So we were able to treat our soldiers and the Germans weren't. And this is why you don't die of things like strep throat or a scraped knee. So the way that most antibiotics work is that they inhibit some necessary process inside of a bacterium. So the idea would be that we are either killing or at least inhibiting the growth of a bacterium by targeting one of their essential metabolic processes. And so some of these processes are listed here: DNA replication, RNA synthesis, translation. So DNA synthesis, RNA synthesis, or protein synthesis are all common targets for antibiotics. And then the question becomes, well, our cells do all of these things. We synthesize DNA, we synthesize RNA, we synthesize proteins. Why do antibiotics not target our cells? Well, because the proteins and enzymes that we use to do these functions are slightly different from those in bacteria. And so antibiotics work by targeting the bacterial version of DNA synthesis or RNA synthesis or protein synthesis, and not the eukaryotic version. But what we can experience sometimes is antibiotic toxicity. So if you prescribe too high of a level of the antibiotics, if they bind all the bacteria and there's a bunch left over, they can bind your compounds as well. So you want to make sure that you, as a physician, do not prescribe too high of a dose of antibiotics, because you can have toxicity where they prefer to bind to the bacterial version because the bacterial version is more complementary to the antibiotic. But sometimes the eukaryotic version is not that different, and they could form a weak bond. So here they're showing you at the bottom some other mechanisms of antibiotic function. So they show you also that some antibiotics can inhibit the cell wall, and some can work against the cell membrane. And then they give you in black examples of classes of drugs or types of drugs that do inhibit some of these processes. So the quinolones, like ciprofloxacin, inhibit DNA gyrase. They can't unwind their DNA, they can't replicate it. RNA polymerase, okay, the ribosome or the mRNA, the cell wall, and the membrane. And then on the other side in green, you see resistance mechanisms. So for resistance mechanisms, we'll talk a little bit more about that, but our major types of resistance mechanisms are we can destroy the antibiotic, so we can make an enzyme to destroy the antibiotic. That's like a beta-lactamase. We can export the antibiotic, right, shown here. We can prevent the antibiotic from getting in. We can try to modify our porins, or we can change or modify the drug target. So bacterial cells, because throughout their evolutionary history they've been fighting with one another, or fungi have been fighting with bacteria, they have to have mechanisms to fight these things off. So even though we talk about antibiotic resistance as being a really important problem in human health, bacteria have had to be resistant to antibiotics throughout their evolutionary history to protect themselves. So it's not something that bacteria are trying to do to make us sick. It's just what bacteria are trying to do to survive. So we have antibiotics that can target the cell membrane and the cell wall. So for example, daptomycin specifically binds to bacterial membranes. I don't know why it says plasmid membrane. It should say plasma membrane, leading to pore formation, depolarization, and death of the cell. So this drug binds to residues that are found only in bacterial cell membranes, causes pores or holes to form in the membrane, which leads to cell death. There are other drugs like polymyxins that target LPS. So these would only work against what kind of cells? Gram-negative. So that's why it's important to know whether or not you're treating a gram-positive or gram-negative infection. So they target LPS, they disrupt the membrane, causing leakage of cell contents and death. And again, the reason that these work against bacteria instead of preferentially targeting our cells, well, we don't have LPS, so that's easy enough. And our membrane composition, even though we both have a phospholipid bilayer, right, our membrane composition is a little bit different. So we take advantage of those differences. And then we target cell wall or peptidoglycan biosynthesis. So the first type of drug that was ever discovered was penicillin. And penicillin belongs to a class of drugs called beta-lactams. They inhibit cross-link formation. So as the cell is trying to build its peptidoglycan, it has to link together the sugar amino acid tail subunits. And this drug, or these drugs, beta-lactams, inhibit that process. They bind to a protein that is called penicillin binding protein. Penicillin binding protein, its function is actually to do the cross-linking. But it was discovered because that's the protein that penicillin interferes with. So penicillin, cephalosporin, and the derivatives, that's what they do. Vancomycin is more of a second generation or third generation cell wall inhibitor. It's a newer drug, but you might remember learning about the escape pathogens, and we do see vancomycin resistance. So vancomycin resistance is on the rise. Vancomycin also binds to one of the precursor molecules and prevents peptidoglycan biosynthesis. Bacitracin is a drug that binds kind of upstream. So it actually prevents the synthesis of peptidoglycan precursor molecules. So it doesn't interfere with directly peptidoglycan synthesis. It interferes with the synthesis of the molecules necessary to make peptidoglycan. And bacitracin, it gets its name because it was discovered in bacillus. So bacillus produces bacitracin to kill other microbes, to protect itself and to kill other microbes. So all bacillus have inherent bacitracin resistance. And we've talked a little bit about the idea that bacteria can share genes with one another. And we're going to talk more about that idea a little bit later. So if bacillus can already have this resistance, what's stopping them from sharing it with other species? And bacillus is really common in all of your probiotic products. So are you consuming stuff that's going to give antibiotic resistance to your own gut? Maybe. Talk to Manny about it. Those of you in Manny's lab, he'll be happy to explain. Okay, so these are our major classes of inhibitors. DNA synthesis, RNA synthesis, protein synthesis, cytoplasmic membrane, and cell wall biosynthesis. Those are the 5 major classes. There are a handful of drugs that also interfere with a particular metabolic pathway for the synthesis of folate, but that's pretty uncommon. So mostly we're looking at those types of drugs, those 5 classes. So then we talk about how we fight microbes now by identifying organisms that produce antimicrobial compounds, purifying those compounds, testing those compounds, marketing those compounds, modifying those compounds so that we can help treat infection. But again, we are trying to fight a battle with organisms that reproduce faster than we ever could. And so it's kind of not fair for us, right? We'll never win. Bacteria can always grow faster than we can. They always can evolve faster than we can. And so the ways that they become antibiotic resistant are what we're going to discuss next. So we can have spontaneous mutations, and we can have antibiotic modification. So when you're thinking about bacteria in a large population, at some point they are going to develop mutations. And it might sound like those of you who are taking genetics, have taken genetics, we talk about that mutation rates are pretty low, mutations aren't very frequent, but if you're talking about a population of billions or hundreds of billions of cells, the possibility of one of them acquiring a mutation that is beneficial is pretty good, better than your odds of winning Powerball. I only play Powerball, this is what Dr. Sukamura told me, only play the lotto if the prize is greater than the odds of winning. So if the odds are 1 in 300 million and the prize is $350 million, you should play. But otherwise, he says don't play. I don't know if he's just trying to take the money. Like he doesn't want me to play because then he could win. So we'll see. We'll see. I'll hit him up if he wins the Powerball. I'll hit him up and let you know what he says. But when you talk about numbers that big, if I bought 300 million lotto tickets, the chance that one of them's a winner is pretty good. If I had enough money to buy 300 million lotto tickets, I probably wouldn't play the lotto, but that's okay. So if you have a population of bacteria that's hundreds of millions or hundreds of billions, the chance that one of them acquires a beneficial mutation is pretty good. In fact, it would be a lower chance that none of them would acquire a beneficial mutation. And so what bacteria can do in order to become antibiotic resistant is they can modify the drug target. They can use an enzyme to inactivate the antibiotic. They can remove the antibiotic from their cell, or they can completely bypass whatever it is that the antibiotic inhibits. So these are the four major ways that microbes become resistant, or the four major mechanisms that they use for resistance. And they can become resistant through random chromosomal mutation, for example. So let's talk about the quinolones. They inhibit DNA gyrase. So that means they bind to a certain spot on DNA gyrase. When they bind to that spot on DNA gyrase is no longer able to unwind DNA. But what if you had a single cell that had acquired a mutation in DNA gyrase that altered the binding site for one of the quinolones, like ciprofloxacin? Remember, we talked about the idea that a single nucleotide change can lead to a single amino acid change, can change the shape or charge of parts of a protein. And so let's say that mutation maybe changed the charge of the place where cipro binds. And now cipro can't bind, but DNA gyrase still works. So a random chromosomal mutation has now conferred resistance to DNA gyrase inhibitors, specifically ciprofloxacin. So all the rest of my friends are killed by cipro, but I'm still here, and I can replicate and now make a population of individuals that are all resistant to cipro. So that's how random chromosomal mutation can lead to resistance. We also see this in proteins or in antibiotics that bind to ribosomal proteins. If you slightly change the sequence of the gene encoding the ribosomal protein, you can confer resistance to that antibiotic. What we talk about a lot is the way that cells can acquire resistance genes from other microorganisms. So these resistance genes can be found on what we call mobile genetic elements. We're usually talking about plasmids, which we've spent some time talking about already. And these can be moved from one microbe to another. So if I have a resistance gene on a plasmid, I can make a copy of that plasmid and I can give it to you because you don't have that plasmid. And now we're both resistant. And then we can give it to more and more and more. And us and all of our descendants are now resistant. So on plasmids, there are genes called beta-lactamases, right? So beta-lactams are penicillin and related drugs that inhibit cell wall biosynthesis. Beta-lactamases are enzymes that cut up beta-lactams. They cut the antibiotics so the antibiotics no longer work. The gene that gives resistance to chloramphenicol adds acetyl groups to chloramphenicol, preventing it from working. So these are mechanisms by which we can share genes that confer resistance. So we've talked about how spontaneous mutation can lead to resistance. We've talked about how inactivation of an antibiotic can lead to resistance. So now we have efflux pumps. So no matter what, for an antibiotic to work, it has to get to its target. If a drug targets DNA gyrase, it has to get into the bacterial cell, get to DNA gyrase, and bind DNA gyrase. Well, I don't want a drug inside of my cell that will inhibit DNA gyrase. So I'm going to take it and throw it back outside. That's what my dad does when spiders are in the house. He doesn't like to kill them, but he doesn't want them in the house. So he picks up the spider and throws it outside. I don't want this drug inside of my cell. Pick it up and throw it outside. And so many, many, many microorganisms have pumps. They're called efflux pumps, and it's basically the way they open the door and shoo that drug outside. So that lowers the concentration of the drug inside the cell. The lower the concentration of drug inside the cell, the less likely it is that that drug will encounter its target. And that allows the cells to survive even if the concentration of drugs is really high on the outside, because they just keep throwing them out. The dangerous thing about these efflux pumps is that they are what we call promiscuous. It's not like you have to have a single pump for every type of drug. Many of these pumps are what we call multi-drug transporters. They're just like, ooh, antibiotic, and throw it out. They don't care what it is. They'll just shoo it out.
So it's, is it any molecule or is it just antibiotic?
They usually work mostly against antibiotics, but they can throw out other toxins as well. So they usually work to remove harmful compounds from the cell. Yeah. And so these can contribute to multi-drug resistance. So there's one called it's MLS, macrolide, lincosamide, and streptogramin. It takes out all three of those kinds of drugs. So Pseudomonas aeruginosa is a bacterium that's highly antibiotic resistant, mostly because it expresses a lot of these multi-drug efflux pumps. And when they're under stress, one of the things that cells will do under antibiotic stress is increase the expression of these pumps. They'll make even more to make sure that that drug gets out of the cell. So they can be resistant by removing the drug from the cell. And also biofilms can contribute to this phenomenon of not allowing the drug to get inside the cell at a high enough concentration. So outside of a biofilm, sometimes you'll see that these cells are super, super sensitive to an antibiotic. But the second you put them in a biofilm, you can prevent access of the antibiotic to the target cells. But also, one of the things they do in a biofilm is upregulate efflux pumps. So they change their gene expression in response to being in a biofilm.
Did efflux pumps just happen to random mutation, or were they already naturally in?
So they already had these types of pumps to remove things, because bacteria are always trying to fight one another, so they had to have a little bit of a defense mechanism. But through constant antibiotic pressure, they evolved to be better. They can evolve to be more of them, and they can share them among species as well. So again, that's another thing that can make biofilms tricky when we're trying to treat human infections. And finally, we can do what we call metabolic bypass. Whatever is being inhibited by the antibiotic, we make it unnecessary. So if the antibiotic is binding to one of my cell wall synthesis proteins, I'll make a different cell wall synthesis protein. I just won't use that one anymore. The antibiotic can bind all it wants, but I've got this new protein that does the same thing and is not binding to the antibiotic. And so I'm sure that the majority of you have heard of MRSA, methicillin-resistant Staph aureus. Methicillin-resistant Staph aureus comes about by bypassing the target of the antibiotic. So methicillin is a beta-lactam. However, it's been engineered. It's what we would call something like semi-synthetic. So chemists took the structure of a beta-lactam, they modified it, And they modified it in such a way that beta-lactamases, the enzyme we would normally use to cut up beta-lactams, no longer work. So cool! We've now made a drug for which there is no resistance. The enzymes won't work against it. It doesn't go into the cell, so it doesn't get pumped out, right, because it's working right there on the cell wall. We've made the perfect drug. Except for the fact that there are now alternative penicillin-binding proteins. So beta-lactam binds to penicillin-binding protein. Oh, shoot, I'm going to die. Oh, wait, I'm MRSA. I have a completely different penicillin-binding protein that beta-lactams can't bind to. I'm going to continue to synthesize my cell wall, and you can just sit over here in the corner. And this is on what we call a cassette, a chromosome cassette. This ends up being another mobile genetic element. These cassettes can copy themselves and pop in and out of the chromosome and can be shared with other species. So that's how methicillin resistance spreads. And so that alternative protein, we call it MECA. It is only made when beta-lactams are being applied. So this is a really unique strategy for antibiotic resistance, and it was completely unexpected. Scientists were shocked when Staph aureus became resistant to methicillin, and then they found out, holy crap, they made their own protein to bypass this perfect drug. Okay, like I said, they've been fighting much longer than us. They'll keep fighting. They fight faster. They fight dirty. It's going to be a struggle. We'll pick up on Friday with persistence and dormancy.